Hello, good afternoon. We will be finishing up lecture 7 today, and we'll have a bit of time left at the end for you to ask questions about either the lab or the upcoming quiz. So let's get started. We still have the last section of lecture 7 to go through, which is about mutation. So let's take a quick note here first. Mutation is basically a permanent change in your DNA sequence. And once you change the DNA, then the instruction to make the protein is going to be altered. And you might end up making a protein that's non-functional, or you might make a protein that ends up uh, working a little bit differently than the original. But mutation in general is it's a permanent change in your DNA sequence. Now we're going to be looking at two different types of mutations, and under each category there are subtypes. So we will be looking at point mutation. And for point mutation, we have what's called base substitution. And then the other kind is called insertion or deletion. Okay, so keep this handy, and we're going to add to it. Okay, permanent change in the DNA of a cell. Okay, so we have a few examples to go through from the PowerPoint slide. I just copied it over here so that it is easier for me to post it as a note afterwards. Right, so over here, let's take a look at the first type of base substitution. Base substitution basically means replace one nucleotide with another. Now, even though the mutation has to happen in the DNA for it to be permanent, I am going to give you an example in terms of what happens after transcription. Okay, so the mutation happens in DNA, and then we're going to look at the transcribed RNA sequence because it's easier for you to understand when you're first learning it. Okay, so keep in mind that the change actually happened in the DNA, but I'm just writing it here as an RNA for you to understand easier, and it's easier to look it up in the codon chart. All right, so we have what's called wild type. Wild type just means normal. Right? So let's say we have UUU as a codon on the RNA. So in the DNA, it would have been like TTT. Okay? But let's focus on the RNA here. And let's say it's mutated. So this is the mutant. It's mutated into UUC. Okay. Please take a moment and look up UUU and UUC on the codon chart and tell me what they code for. What amino acid does it code for? Anybody? That's right. Thank you. So UUU, if you look it up, UUU, it codes for PHE. And then UUC also codes for PHE. So in this case, changing the codon does not change the amino acid. So you wouldn't even know about it. There is going to be no effect on the protein sequence. That's why this is called silent mutation, okay, or neutral mutation. If you're going to have a mutation, you better hope that this is this kind of mutation because it's not going to have any negative effect on you. Is that okay? It's pretty straightforward so far. Let's do another example. We are still doing base substitution. Remember back at the beginning, mutation, we are learning what's called point mutation. It happens at a single location. And there are two different types, base substitution or insertion deletion. We're still talking about base substitution. So here we have our wild type again. Let's say we have ACU. ACU. Then we're going to have a mutant, which will be CCU. Right, so I am only changing one nucleotide at a time. I probably should highlight the previous one as well. UUU becomes UUC. Okay, please look it up. What is ACU and what is CCU? So ACU is THR. And then afterwards, CCU is pro, right? So in this case, changing the codon results in a different amino acid, right? This is called missense mutation. It's a mistake, basically. Missense mutation. Now, you might wonder, is this bad? Well, the effects depend on where the mutation is in the protein. Right? So we have different kinds of proteins. Some proteins are for structures, some proteins are for transport, some proteins are for, chemical reactions. They're enzymes. So you might remember our discussion on enzyme, right? So an enzyme is usually going to have, well, not usually, they always do. They have what's called an active site. Okay, this is an enzyme. And the active site is where you will fit the reactants in, right? The substrate. And this is going to go in right here, right? So there's a key and log relationship between the shape of the active site and the shape of the ingredients. Now, if you change one of the amino acid near the active site, then it's possible you alter the shape of the active site and then the enzyme no longer works. So that could be a big effect. Whereas if you have a mutation that's like elsewhere, let's say right here, right, somewhere away from the active site of the protein, then maybe it's okay. Maybe it's not as big of a problem, right? So it's really context dependent. It depends on the protein and depends where in the protein it happens. Now, all the way back in lecture two, I showed you a picture of, you know what? I probably still have that picture. I showed you a picture of amino acids. Okay, and you can see you can group amino acids based on their properties. Some amino acids are nonpolar, some are charged, positive charge, some are negatively charged, some are polar. So if the mutation, if the missense mutation changes the amino acid from one nonpolar amino acid to another nonpolar amino acid, then maybe it's okay, right? Because they are similar enough in terms of property, it might not cause as big of a problem. Whereas if you change the amino acid from a nonpolar to a polar amino acid, then that could be quite a bit of a problem, right? So again, to evaluate whether the mutation is good or bad, it depends on a lot of things. It depends on where it happens. It depends on what kind of amino acid are you switching to, and it depends on the kind of protein that it is happening to. Think of it like this. Like if I say, there is a nail in your car, like a long piece of nail stuck in your car, is that going to be a big problem for driving your car? What do you think? Okay. Okay, like I said, yes, this is a big problem, right? Yeah, I mean it could be, right? If the nail is stuck in your tires, right? Then it's gonna give you a flat tire eventually, if not immediately, then you can't drive your car anymore, right? But if the nail is stuck like at the door or something, like it's not gonna look good, it damages your car, but it's not gonna affect your car's function, right? So the mutation is kind of like that. It really depends on where it happens in the protein. Okay, it could be debilitating, completely cause the protein to lose its function. So that would be an example of a loss of function mutation. And if it happens in the tumor suppressor gene, then that's a big problem, right? The cell cycle can become uncontrolled. And if it's a loss of function mutation in a proto-onco gene, then maybe it's not as big of a problem, right? The red lights and the green lights that we talked about last time. All right, so that's missense mutation. Are we together, guys? Everybody with me? Not a lot of people today, actually, but that's okay. Last one is nonsense mutation. So let's say we have a wild type here one more time. Wild type. And we will say this is CAG. And we're gonna have a mutant that is UAG. Now, I want to emphasize, even though we're doing RNA here, the change has to happen at the DNA level. Okay, I'm just doing the RNA transcript to make it easier for you to follow. So CAG, CAG, that's going to be GLN, GLN, and UAG. You should remember UAG is one of the stop codon. Yeah, I know the I and the L looks the same, right? But the I is they are all lowercase, so I and L. The confusing one is this one. This is ILE. Okay. Anyway, so here it goes from GLN to stop codon, right? Which means if you have like a bunch of codons, okay, don't I'm not gonna write it down all of it. So starting with AUG, blah 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 blah, and then at the end, let's say we have a stop codon here, right? So normally you would start here and then you would make all the connect all the amino acids until you get to the stop codon. But let's say you have this nonsense mutation and you introduce a stop codon here. So instead of CAG, this one mutates to UAG, and so when you build the amino acid chain, when you build the protein, So you have amino acid, amino acid, amino acid, amino acid. Instead of having GLN next, you're gonna stop, right? So what happens? Right? This causes the translation process to stop prematurely. Okay. We're not supposed to stop. We still have like five, six, seven more amino acid to build, but the nonsense mutation caused it to stop. So it creates, okay, creating a truncated protein. Truncated means shortened. Truncated protein. All right, I know you guys are like super quiet, but let's try to discuss about this a little bit. Is this going to be a problem? Is this bad, in other words? Okay. Some people say thing is bad. Anyone else? It's nonsense mutation bad. Is it worse than missense? No. All right. So the answer is it depends. Okay, it depends. Depends where the nonsense mutation happens. If it happens earlier on, yeah, it's pretty bad. You're going to be losing, in this case, two-thirds of the protein. Protein's probably not going to even be working. Okay. Uh, but if the mutation happens towards the end, like if you have the mutation like over here, then maybe it's okay. Maybe it's not as detrimental. It's not as bad as happening earlier on, because then now you're just missing maybe two or three amino acids. Right? So for nonsense mutations, the later it happens in the gene, the less likely is going to have an impact. Okay, I'm not saying there is going to be no impact, but there is a lower chance of having a negative effect. So are we together? Neutral, missense, and nonsense. I certainly hope so. So we're gonna go over here. I'm gonna give you a wild type. Wild type. That's what they're called. We will say it is um UCG. UCG. Let me see if this is gonna work. Yeah, totally just making this up on the spot. So I want you to create three different mutations. I want a neutral, missense, and nonsense. All right, go for it. Please send it to me when you have it, okay? Okay, that was about 4 minutes. Does anybody have an answer they want to share? Okay, thank you, Fahima. So UCC for neutral. Okay. I mean, there are more than one correct answer, by the way. So the most important thing is to check what it becomes. So UCG is SER. If you mutate it to UCC, it is still SER. Okay, so that's good. For missense, yeah, or it could be like UCA, UCU, UC, yeah, any of them would be okay. All right. Now, if you want it to be missense, your best bet is to change either the first one or the second one. Okay, I'm going to change the first one. Change it to whatever you like. A, C, G, and then you're going to just check it out. A, C, G, which is THR. So that's a missense. And then for nonsense, you're going to change, I'm going to change the middle one. Okay, so that's going to be stop. All right, so last time, maybe it was last time, I don't remember. Like last week, I told you there are 64 codons, but only 20 amino acids. So there is a lot of like redundancy here, right? And so PHE is coded by two things here. SER is coded by four things. Pro is coded by, four things here. So basically, these redundancies serve as a protective mechanism against mutation. So even if you have a mutation, because we have so many redundant codes, you might just end up having a neutral mutation, and it's not going to harm you um, at all. All right. Now, don't get me wrong, not all mutations are necessarily neutral or bad. You can have mutations that gives you a survival advantage. For example, there are mutations that make people immune to HIV. Okay? There are also mutations that would cause a person to be um, immune to what is it, malaria. Okay, they cannot get malaria. The disease that's spread by mosquito, right? So there are mutations that out there that are beneficial for certain, but most of the time, mutations have negative effects. Okay, so that's that. Now we're gonna talk about insertion and deletion now. So for insertion or deletion, we have two consequences. Okay. Insertion deletion is exactly what it sounds like. You are inserting extra nucleotides or you're deleting nucleotides from the sequence. So if you are inserting or deleting nucleotides that are multiples of three, then you will end up having extra or missing amino acids. If you delete three codons, sorry, if you delete three nucleotides, rather, if you delete three nucleotides, then you end up missing one amino acid. If you delete six nucleotides, then you will be missing two amino acids. Okay. On the other hand, if it's not multiples of three, if you're inserting one extra or if you're deleting two, then it's going to lead to something called frame shift. Okay, and frame shift is going to be uh, extensive, extensive missense, okay, and or nonsense. I'll explain. Okay, just copy it down first, and then I will explain it to you. Okay, let's go back to the PowerPoint slides for a second. This slide just shows you the different kind of mutations, right, happening in the DNA level. and then it will get carried onto the RNA when you do transcription. So you just, read through the examples yourself to see. This is sickle cell anemia. Okay, so sickle cell anemia is a condition where your normal red cells become sickle shape, okay, or like a moon shape, if you would. And it's caused by a missense mutation in hemoglobin. So hemoglobin, which we actually learned previously in lecture 2 when we talk about quaternary structure. Hemoglobin is a protein that transports oxygen and carbon dioxide in the blood for you. They're found inside the blood cells, and it has four polypeptides in it, and one of the chains has 128, I think, amino acids in it, and the first couple ones are shown here. And so, in the normal hemoglobin sequence, this amino acid is supposed to be glutamic acid, which is a polar amino acid, and the GAG codon that codes for it is going to get mutated into GTG, right? So if you go and do the transcription, it's going to become GUG, which codes for valine. And valine is nonpolar. So in this case, you are switching from a polar to a nonpolar amino acid. And even though the change is just a single amino acid, it's enough to cause the hemoglobin to misfold, and which ultimately cause your red cells to become sickle shaped. Okay? People with sickle cell anemia cannot deliver oxygen throughout the body efficiently, so they are anemic, they have shortness of breath, they feel tired, and these red cells can get stuck in the blood vessels and cause quite a bit of pain as well. So, there was no cure for sickle cell anemia because people are born with it as a result of mutation. And what happens now is they are doing a gene therapy to try to fix this. Okay, we'll learn more about gene therapy later on in the course, but basically, if you can deliver the correct version of the gene to replace the faulty one, then your body's going to start to make normal hemoglobin again, and then your red cells are going to be normal once again. Okay. And I think there are a few successful cases of gene therapy um, in the past year or so. Okay, so insertion deletion, right? If you are inserting extra or deleting nucleotides that are not a multiple of 3, then it's going to lead to a frameshift mutation. Let's try to understand what frameshift mutation is by looking at this example here. All right, so let's suppose we can only read 3 letters at a time. Okay, so you can read the sentence here. It says the big cat saw the fat dog, right? But what happens if I delete the B here? Right? If you delete the B, then the sentence is going to be the, which is no change. But instead of BIG, the next thing I read will be IGC. Remember, we can only read in triplets. So everything else after the deletion is going to be gibberish, right? It's not same as the original sentence, and I don't understand it anymore. What if I delete 3 letters? If I delete the whole word big here, then the sentence structure is preserved. The cat saw the fat dog. I still understand what I'm reading, but I'm missing an adjective. Okay, maybe it's important to have that adjective, but maybe not, right? Again, it's context dependent. If you add an extra letter, add an extra letter right here, the K, then we have TH instead of THE, now it's THK. And again, everything after that point is not going to make sense anymore. Right? So these 2 examples, they are frame shift because we are shifting the reading frame. So the ribosome, like us, can only read nucleotides in triplets because 3 nucleotides makes up a codon, right? So if you shift the reading frame, everything's going to be wrong after that. In the last example here, you inserted the word red, which is a multiple of 3, so that's fine. Sentence structure is preserved. Now we have an extra word there, I mean, it depends what you insert too, right? Sometimes if you insert something totally weird, like instead of inserting the word red, you insert the word mud or something. Okay, so the big mud cat, like that doesn't make sense anymore, right? So you might change the meaning of the sentence as well. And that's also what we have to consider when we add have insertion mutation. Does the extra amino acid change the overall sequence significantly? All right, so like if there's a section that's made up of a bunch of polar amino acid and then you add an extra one in there, then maybe it's okay. Right? Maybe it's not gonna cause huge problem. But if you insert a non-polar in there all of a sudden, then it could cause more problem, right? So let's do some practice together. This is in your study guide, which I asked you to print. If you don't have it, I guess it's okay. You just have to copy what's on the screen. This here is the template DNA. Right? So what I need you to do is to change it into mRNA by transcription and then find the amino acid sequence. All right. So I'm just going to chop them up into little triplets so it's easier for you to not, uh, get lost. Sometimes you misread the letters and you shift a spot and everything's wrong, right? Even I make that mistake sometimes. All right, so here this is the template strand. How do you make the mRNA sequence? Do you remember from last class? Do I copy it and change the T to U, or do I do base pairing? What's the correct method here? If I have the template strand, how do I get the mRNA? Copy or base pairing? Cheyenne said base pairing. That would be correct. Okay, very good. So please take a moment and do that and write the amino acid sequence as well. Your quiz is going to be this Friday and Saturday. You can do it on either day. It doesn't matter to me. Make sure you print a codon chart for the test. Okay. I strongly don't recommend you using your phone for the codon chart. It's really hard to, scroll back and forth and stuff like that. Just print it out. It's much easier that way. All right, so please do this. I give you like, 2, 3 minutes. It shouldn't take you too long. Okay, go for it. Okay, I hope you had a chance to finish that. So we will do complementary bases here. Please double check my work. And if you go ahead and look up the amino acids, it will be like this. Hey, check your work against mine. Let me know if there is a particular one that you disagree, and then I can explain it. Okay? Give me some sign that you are done checking, and then I'll move on. Like, maybe like a thumbs up or something like that. Okay, I got a few confirmation. That's good. That's good. All right. So let's take a look at here. This is a mutated sequence. And what you want to do here is to chop them up into triplets. Again, that's like the first thing you do. Chop them up. And you want to check the letters one by one and see where the mutation is. Do you see the mutation? Is it in, codon 1, 2, 3, 4, 5, 6? Where is it? That's right. Okay, so in codon number 2, in the wild type, it is ACC, but now it is ATC. So there is your mutation. And remember, the mutation happens in the DNA, right? And so when you do the transcription, TAC is still going to be AUG, and then ATC will now be UAG, and the rest would be the same as before. Okay, I'll give you a moment to copy that down. This is the change. Oops, wrong color. This is the change. The rest are just the same as before. So let's do the translation. AUG, you should have memorized it by now. It is the start codon which codes for MET. What about UAG? What is UAG? Here, I'll write underneath here. What is UAG? You should also remember UAG. Yeah, it stopped. That's right. And then don't keep going, okay? Because once you stop, you stop. What kind of mutation is this? We'll talk about the effect in a second, but how would you name this mutation? That's right. This is nonsense, right? Caused by a base substitution. Base substitution, and then nonsense. Will there likely be effects? Absolutely, right? Because like, where is the protein? It's not there, right? It's just one amino acid floating around. It's like the protein is gone. So yes, the protein is not made, right? You're basically not making the protein. So lost of function. If this was a red light, if this was a tumor suppressor gene, then you're gonna lose the red light. Hopefully you have a backup that's still working and it will be fine. But that's how the concepts are connected together, right? Okay, let's try that again. Number two, I'll do this one with you as well. We're gonna chop them up into triplets. We always do. And again, this is in your study guide. Now, when you chalk them up into triplets and you notice, hey, well, there's an extra one. I can divide them equally. That's a telltale sign that this is an insertion mutation, and it's probably you inserted an extra nucleotide. So you're gonna have to compare to the original once again and see where the extra nucleotide was inserted. Is it codon 1, 2, 3, 4, 5, 6? Where is it? Find it. That's right. It's at number 2, codon number 2. It was TAC, so TAC, that's fine. And where it was ACC before, now is GAC. And this was ACC, but now there's an extra G inserted. Insertion. Okay, we will transcribe the thing using base pairing rule. AUG, GAC is going to become CUG, so on and so forth. All right, and if you do the translation, it's going to be MET, LEU. Please check your answers. What kind of mutation is this? Oh, it's an insertion. And because we did not insert multiples of 3, it resulted in a frame shift mutation. And basically everything after the point of insertion are wrong. They are all missense, essentially. So extensive. Extensive means like a lot of it, right? Extensive missense mutations. Will there likely be effects? What do you guys think? Yes or no? Yeah, that's right. I agree. Yes, effects. Because the sequence is completely different. Like, I don't even know if this protein is going to do anything. And if it does something, for sure, it's not going to be the same function as the original. Okay. It's like the recipe has completely changed. You try to make a cake and you end up making, I don't know, roast pot or something, right? Totally different. Right? And moreover, the original stop codon has been missed. So it's going to go on for some time. It will stop eventually if it hits another stop codon or if it gets like too long. It will stop eventually. But we missed the original stop. Okay, I'm going to give you about 5 minutes to do the next one on your own. And then we're going to move on. Just let me know if you need help. Okay. Hey, it's about time. What kind of mutation is this? Anybody figured it out? Have you guys find? Yeah, neutral mutation. That's correct. Okay, so this one here, if you try to find the mutation, it's actually right here. Right? Like you just compare them one by one. Instead of TTG, it's TTA. So to speed things up a little bit, everything before and after are going to be the same. If you do the TTA, that's going to be AAU. And if you look up AAU, it is going to be ASN. So it was ASN before, it is still ASN. So in this case, it will have no effects. It's a base substitution. And it result in neutral mutation. Okay. Do you guys want to try another one, or are we good for now? Any questions? Okay. Anyone want to do another one? Yeah. Yeah, let's just do one more just for good measures, okay? Here it is. Give me a moment. I will copy the original for you. And there is the original. Okay. Please try number 4. Okay, that was another 5 minutes. What have you guys found for this one? Where's the mutation? Mhm. So instead of ACG here, it becomes ACT. Everything else is the same. You can transcribe them later on, but the important thing is this one's going to be UGA, which means it's going to be a stop codon. Okay. So it's a base substitution, and then it's going to be nonsense. Now, will there likely be an effect? I guess you can say yes because you lose an amino acid, right? You can say maybe. I don't know. It's it's these are too hypothetical. Okay, maybe. Okay, only lost one amino acid. Okay. Yeah, so maybe it's just minimal effect. However, you can also argue this protein only has one, two, three, four, five amino acids. So if you lose one amino acid, that's like 20% of the protein gone, right? So, it could be pretty bad, right? Compared to something that's like 100 amino acid long and then you lose one and you still have 99% of the protein there, right? So, you know, it's gonna be context dependent. That's what I'm trying to say. Okay, you can do number 5 on your own later. All right, moving on. We just gotta cut the break and finish up. We're almost done anyways. We have 4 more slides, and I'm gonna copy them here. Okay, so now we're gonna try to excuse me. We're gonna try to create our own mutation. Create a base substitution in the coding strand below that will result in a missense mutation. Right, so this is coding. It tells you in the question. And this is DNA, right? So you can pick any codon. You don't have to do the whole thing. You can just pick, let's say you pick this one right here, okay? CAG. So if you go ahead and do the transcription, because this is coding, I can just copy directly for the mRNA. So it's just CAG is the same thing. So let's look up what CAG is. CAG is GLN. And this is the amino acid. And if I want a missense mutation, you just change the first one or the second one. Usually it works. You can change it to GAG. So this would be GAG as well. And if you look up GAG, it's going to be GLU. So that's an example of how to do the question. Do you have to use the second one? No, not really. You can choose another one. Let's say I choose this one here. Okay. So AAT, it will become AAU. Let's look up AAU. AAU is ASN. So let's change the second one here to C. So ACU now. ACU is Stironi. So that's how you do it. You don't have to do both, right? You only have to pick one and create a mutation, and that's it. Okay, you try. This is the coding strand. And we want to do a base substitution, which means you're replacing a nucleotide. and you want to create a nonsense mutation. For nonsense mutation, we need to have a stop codon, which would be UGA, UAA, or UAG. But these are on the RNA, and we want to do it on the coding strand. So how do you convert these into coding? What's UGA going to look like on the coding strand? The relationship between the coding and the stop codon, sorry, the coding strand and the mRNA is that they are going to be the same. So instead of UGA on the coding strand, it's going to be TGA. Right? UAA is TAA and UAG is TAG. So basically, what you need to do is you need to look at the coding sequence and you see which one you can change so that you can create a stop codon. For example, over here, we see the codon CAG, which if I change the C to a T, then I have a stop codon, right? If you do transcription, then it's going to become UAG, and then it's going to be stop. Is there another location I can create a nonsense mutation? Nonsense mutation is a little bit different from missense. It's not always possible for every codon, but do you see another spot? Anybody? Okay, Michael, this is AAG. How would you change it? You change it to, that's right. So you can change the first one to a T. So that's going to be UAG again, and then that will also be stop. Okay, any questions? All right, let's do number 3 here. This is the template strand that we're given this time. And we're also trying to create a nonsense mutation using base substitution. So let's write down the stop codon again. UGA, UAA, and UAG. This is the mRNA. And so on the template, it is going to be complementary to these codons. So instead of UGA, it will be ACT, ATT, ATC. So all you have to do now is find a codon where I can change one of the letters to create these stop codons. Do you see where it can be done? Hmm. Yeah. So AAT here, you can totally change it to ATT. Right? I don't think there is there is another spot here. You can change that to A, right? So just one of those, right? You don't have to do both. Because if it stopped here, it's not going to go on, right? So that's example three. Last example. So like on the quiz, I'm not going to be able to ask you to create mutations like these because physically can't do it on the computer, right? But practicing these things will help you understand more about mutation and probably will help you answer the questions a little bit better. Create an insertion mutation in the coding strand below that will result in a truncated protein. So truncated protein means shortened protein. I need a nonsense mutation again. Okay? And I'm just gonna copy from here. Gonna bring it all the way down. So I need to insert something into the sequence to create one of these stop codon. Right? So ATG, GAA, but instead of GAA, if I put a T here, then the codon will be ATG and then TGA. Right? TGA, of course, it's going to get transcribed into UGA, which is going to be stop. Okay. Another place you could do it at would be right here. You can insert a T. So ATG, GAA, TTC, TAA. TAA is going to get transcribed into UAA, which is, of course, also a stop codon. So you can create nonsense mutations in different ways. You can create it by base substitution, where you're just changing one nucleotide, or you can be creating a nonsense mutation through insertion or deletion, where the new reading frame has a stop codon in it. Yes, Micah.
I don't understand how you change the G to a T. Wouldn't it
You over here, you mean?
yeah.
Yeah, we
Wouldn't you
didn't
have to change it to a C
yeah.
instead?
Okay. So first of all, this is a coding strand and from coding to mRNA,
Hmm.
it is just direct copying, right? So, the A is still going to be A, the C is going to be A, G is going to be G,
Hmm.
but the T is going to be U, right? You with me on that?
Um,
Okay. Coding. If coding is A, C, T, G, the
hmm.
mRNA will be A, C, U, G.
Okay.
Because we just copy, right? Coding and copy, right? We said. But there is no uracil on the mRNA. Sorry, there is no thymine in the U on the RNA. That's why we have to change it to uracil, right?
Okay.
Okay. But that doesn't answer your question. Over here, we are not changing the G to T. Okay, changing it would be like this, like crossing it out and then replacing it.
Hmm.
Over here, we are doing an insertion mutation. So I'm adding an extra T here, right? So when you add the extra T, when we do the transcription, it will be AUG for the first one.
Hmm.
And then instead of GAA, now it'll be UGA. And then the next one is going to pick up from here, which is AUU, so on and so forth, right? The rest doesn't really matter because after the AUG, we've reached a stop codon. The stop codon wasn't there before, but by inserting an extra T, we've created a stop codon in the new reading frame. And that's how it
Um,
becomes a nonsense.
I'm still not understanding because for T. You said there's no T in the mRNA. So
Yeah,
wouldn't you change it to an A instead of a U?
yeah, but this is
How
the
you put the
DNA
first?
though, right? The coding is the DNA, right? Okay, how about we try this instead? Right? That's okay. Um, can you change it to mRNA for me, please?
So it'd be U.
Okay, but this is the coding strand, right? So for the
Oh,
coding strand, we do not do base pairing. For the coding strand, we
mm-hmm.
just copy directly, right? So go ahead, please.
It would be would it be A
Mm-hmm.
T
Okay.
G
I'm sorry, what was the second one?
A U G
Good. And then
C.
again, this is
Uh,
the coding strand. We do not flip them. We do not do base pairing. If it's coding, we just copy directly. Okay. So GAA will still be.
G
Mm-hmm,
A
mm-hmm.
A
Yeah. Carry on.
E T C.
Okay, but
No,
we
wait. Uh, U U C
good. And then
A A U
very good.
G A U
Yeah. And then
U A A.
that's it, right? Yeah, so we just copy, but we switch the T's to U's, right?
Oh, okay, okay, I'm
Yeah.
understanding now.
So now we have to create a mutation. So what we're gonna do is we're gonna add an extra U here to create a UGA, which would be the stop codon, right?
Um, yes.
Okay, because the UGA, UAA, UAG, these are all stop codons, right, over here.
Mhm.
But we must reflect the change in our DNA sequence. So if you want to have an extra U here in the mRNA, what letter must we add in the original coding sequence
Um, so that it goes, so that it's a stop codon.
so that it's going to become a U when you change it to mRNA? What letter does the U correspond to in the coding DNA?
A.
Okay, so once again, right, for the coding strand and the mRNA, the relationship is that they are the same, right? We do not have to do the base pairing rule. So it wouldn't be an A, it would be just T. Okay? If this was a template strand, then it would have been an A because template strand and mRNA, they are complementary to each other. Is that okay?
Yep.
All right. Just, sit on it for a little bit more. If you have questions, please email me. Okay, and that's it for the mutation lesson. All right. Now, no class on Thursday. Just take the time to finish your lab. And then, like I said, the quiz will be available to you on Friday and Saturday. We'll resume Zoom class next Tuesday. Now, if you want to go, go. Okay, I'm done teaching. But before you go, do you have any question about the lab or the upcoming quiz? Anything you want to ask me at all? Have people had a chance to work on the lab yet? Not yet. Okay. Yeah. So there are some resources that I posted online on the did I post it? Let me just double check. I think I did. Yeah, over here, right? There is a video. Oh, there is a video that shows you how to use the codon chart if you're still a little bit unsure. In terms of the lab, you just complete it and then you submit it in the submission folder by is this your class? You guys are 202, right? Okay, so I think I got the wrong date here. It is going to be due on Thursday. So November 14th instead. There is a help video for the calculation part, which people struggle a little bit with. So make sure you watch that. And for the quiz, 30 multiple choice questions, 60 minutes should be more than enough. Okay, like that's 2 minutes a question. Okay, so don't come on Thursday, just finish up the lab. No practice quiz, just do study guide, and there are some self quizzes that you can do as well, right? In the assessment folder. So we good? Nothing else before I sign off? Okay. Oh, and one more thing. I don't know if I posted yet, but if you go to test review, I have not posted yet, but I will do it. Actually, I'll do it right now. Just hang on. I'll show you what I'm posting. The lecture 6 and 7 concept check questions. Okay, so like the poll questions that we've done in class, sometimes I skip through it because like either people are not doing the poll or we don't have time. But the answers are here for lecture 6 and 7. Right? So if you need to go through those yourself, please check out the file. Okay, that's it for now. Have a good rest of your week. I'll see you next Tuesday. Bye bye.